Hello everyone. Welcome to today's lecture. In this lecture, we will learn the fundamental concept of culture media used for microbial cultivation. Cultivating microorganisms is an essential process in laboratories for observation and research, allowing us to study microbial growth, characteristic, and metabolism. Therefore, understanding culture media is crucial for effectively growing microorganisms. In this lecture, we will first define what culture media are. Then, we will learn about the different types of culture media and how to prepare them for microbial cultivation. Culture media are mixtures containing various nutrition necessary for microbial growth. The composition of media varies depending on their type of microorganism, cultivation conditions, and experimental purpose. Basic components of culture media include carbon source, nitrogen source, mineral, and growth factor. Culture media can be classified based on various criteria. Firstly, they can be categorized by their physical state: liquid media, solid media, and semi-solid media. Secondly, media can be classified by chemical composition into two types: defined media, where the exact chemical composition is known, and complex media, where the exact composition is not fully known but contains ingredients like extract or digest. Lastly, culture media can be classified based on their purpose. Selective media is designed to isolate specific microorganisms from mixed culture, while differential media helps to distinguish microorganisms based on visible characteristics such as color changes in the medium. As mentioned earlier, culture media can be categorized into three types based on physical state: liquid, solid, and semi-solid media. Liquid media are liquid form and are prepared by dissolving powdered media in distilled water, followed by autoclave sterilization. A commonly used liquid medium is LB broth. Solid media are prepared by adding agar to liquid media, followed by autoclave sterilization. The media are then poured into petri dishes and solidified. Solid media are useful for observing and isolating microbial colonies. A representative solid medium is TSA, tryptic soy agar. Semi-solid media are prepared similarly to solid media, but contain a lower concentration of agar powder, allowing them to maintain a semi-solid state, solid at room temperature, but turning liquid at higher temperatures. Semi-solid media are commonly used in bacteriophage experiments. A well-known example is MTM, used to assess microbial motility. Next, we will learn how to prepare two commonly used solid media: TSB agar and LB agar. The materials used in this experiment are as follows. TSB powder is used for TSB agar, and LB powder is used for LB agar. Finally, let's go over the experimental procedure for preparing each medium. To make 1 liter of TSB medium, you will need 30 grams of TSB powder. Therefore, to prepare 100 milliliters of TSP agar, start by adding 3 grams of TSP powder to an Erlenmeyer flask. Next, add 1.5 grams of agar powder to the flask to solidify the medium. Then, use a graduated cylinder to adjust the volume to 100 milliliter with distilled water. Mix well using a magnetic bar and stir. Cover the openings of the flask with aluminum foil and sterilize it using an autoclave. After sterilization, pour the medium into petri dishes and allow it to cool and solidify. Be sure to calculate the correct amount of medium for each petri dish, which should contain about 15 to 20 milliliter. To prepare 100 milliliters of LB agar, use 2.5 grams of LB powder and 1.5 grams of agar powder. The rest of the procedure is the same as for the TSP agar preparation. In this lecture, we have learned about concept of culture media for microbial growth, the different types of media, and the process of preparing this media in the laboratory. Understanding how to prepare and use media is essential for successful microbiological experiments. With this knowledge, you will be better equipped to work with microbial cultures in future studies and research. Thank you for your attention.